So really thank the meeting organizer to invite me over. It's such a pleasure to hear those exciting research on pediatrics tumors. And so today I will talk about some ongoing work on this exciting project of proteomics genomic landscape of high-grade glioma, but spreading on pediatrics, adolescent, and young adults. As Harry already mentioned, in the past few years between CBTAC and KISS and the CBTN, we have had some wonderful collaboration and applied the proteomics genomics technology to study pediatrics brain tumor already. And under CBTAC, we also have the parallel adult GBM study that has been kind of published 2 years ago, last year. And in the coming CBTAC 4, actually, there are many exciting new projects ongoing or has been planned, including more proteomics genomic study for pediatrics brain tumor, which will actually will be a collaboration with Hui and many other CBTN investigators. And also on today's talk, I will focus on this ongoing project, which is using proteomics genomics strategy to study high-grade glioma, but we want to extend the age span from the pediatrics to the adult with a focus on the adolescent and the young adult age range. Okay, so this is today's focus. I will be brief here. I think most of the audience here probably be very familiar with the pediatrics and AVA high-grade glioma. So as I mentioned, we are interested in this age span of 15 to 40. On one hand, those patients actually has been greatly understudied and underserved. On the other hand, it has been suggested that the HGG and the glioma from this age very likely have unique biology. To illustrate that, so Dimitri, our team member at Sinai, created an in-house database of HGG and the GBM data, which including the survival outcome of patient and the accompanying mutation profiles of the patient from some of the recent HGG-GBM studies in the literature. Based on that database, if we look at the survival of those patients from different age group, we can clearly see that the patient from this adolescent and young adult age group has significantly better survival than the adult and the pediatrics age group. Like this 3-year survival is almost, survival rate is almost doubled in this young adult group, AVA group. So this is quite intriguing, right? One possibility is that they may have very different type of tumor. So that is the reason that they have very different outcome. Or another possibility could be that maybe the tumor intrinsically is similar, but because of the brain development is at different stage and the interplay between the brain development and the tumor kind of can help those AVA patients to do better. If that's the case, that if we can understand that interplay, maybe we will have better, more creative treatment for the GBM and HGG across all the age span. Okay. So those are some intriguing questions we want to understand. And at the same time, if we look at the mutation profiles of all the HGG-GBM patients, we can clearly see there are 3 groups of genes with very different mutation frequency along age. Especially, we focus on this first group of genes, including TP53, IDH1, ATRX. They really have much higher mutation frequency during this AVA age span. So this is another kind of evidence seems to suggest the AVA HGG most likely have very unique biology. So to better understand this unique aspect of that age group, the leadership from NCI, CBTAC, Harry, Tara, together with the leadership from CBTN, Adam, Brian, and others, successfully put together this great collaborative project to using the proteomics genomics technology analysis to understand pediatrics and AVA HGG tumors. And we also call this study HOPE study. Of course, this is a good name. Okay, so this is a cohort of the HOPE study that we are focusing on. So we have 95 fresh frozen tumors collected from 87 patients. And among those tumors, about 2/3 is actually grade 4 glioma, while about 1/3 is grade 3 glioma. And also about 40% of the tumors actually is from the AVA age span, while 60% of the tumors is, the patient actually is coming from the pediatrics age span. So we applied proteomics, phosphoproteomics, whole genome sequencing, RNA-seq, and methylation analysis platforms on each of the tumors. And this figure kind of summarized the availability of different modality of the data on each of the tumor sample. And at the same time, we have planned or has already carried out some of the validation experiment using either targeted mass spec-based platforms or single cell RNA or microRNA that we can leverage to try to validate some of the observations we observed in the discovery data set. Okay, so that is the scope of the study. So this figure shows the oncoprint of this whole cohort. Well, in general, pediatrics tumor has low tumor mutation burden, as we can obviously see. There are quite a few genes, and actually cancer genes has showed very frequent mutation in this cohort, including this TP53, H3F3A, and the ATRX, which is very consistent as the mutation pattern we see in the bigger reference database that I showed earlier. Okay, again, the interest here is really trying to understand the molecular characteristics that was driven by their tumor activity. But we know there's a big player in this AVA HGG and the pediatric HGG is that the brain really experienced multiple developmental sessions from pediatrics to adolescent and to young adult. And those developmental factors really greatly influenced molecular characteristics. And we know there probably will be important interplay between tumor activity and brain development. So the goal of this project is really dissect those complicated relationships and trying to identify the key molecular pathway activities and markers that can explain the tumor activity, and that really can predict the patient outcome. Okay, so that is the goal. To tackle that challenge, we utilized an analysis strategy that we try to decompose the molecular profiles into two parts. One part trying to characterize the developmental process influences, and the other part is kind of like a more orthogonal space and trying to look at the intrinsic tumor activities that is not influenced by the developmental functions. So to do this, I will using one gene as an example. So so this figure is showing the protein abundance of IDH2. The x-axis is the age of the patient. The y-axis is the protein abundance of IDH2 in each individual patient. And we can see, although there are large individual heterogeneity, but if we fit a population kind of mean curve using functional analysis, we can clearly see that this IDH2 has a trajectory which increased with time in the adolescent, and then seems, decreased with time after adulthood. And so this is an interesting way to summarize the data that we call this trajectory, this population trajectory changing of time as a developmental trajectory in tumor tissue, DTT. And then for each tumor, we also will be able to look at the orthogonal part, which is the residual of the abundance. That is basically the abundance measurement of this protein in this particular sample to the population level trajectory. And we call that as the residual developmental trajectory information. So we will then using this two components. One reflects the developmental process in the tumor tissue, and the other reflects the orthogonal part in the tumor tissue, trying to understand those tumors. Okay, let's first focus on the developmental process. And not surprisingly, actually, for majority of the RNA and the protein and the phospho, we see stronger dependence of their abundances on the age. So this heat map is really illustrating the estimated population level, the DTT matrix based on different omics data. And based on the striking structure, we are able to group patients into 4 clusters here. And while the conventional kind of age cutoff of 15 and 40 are nicely supported by this clustering, we found another more kind of substantial striking breakpoint at age 26 that can be seen in both the heat map as well as the left-hand kind of summary of the consensus clustering when we vary the cluster number K. Okay, so another piece of information is this panel on the top, where we are leveraging the big reference cohort that I mentioned earlier, trying to see what are the survival outcomes for patients at a different age span. So using that cohort, we are able to estimate this kind of smoothed survival days function. And we can clearly see that the patient coming from this young adult group between 26 and 40 has the best survival compared to the other age group. And this is further confirmed using a Cox regression model that the young adult age group has significantly better survival than the adolescent. And the adolescent has significantly better survival than adult and pediatrics. So this is a nice way to using the molecular profile, the trajectory, to gain some insight of those group of patients. And further dive into proteomics data. So here, this heat map is only showing the proteomics data. So now we can see more details. The proteins actually here can be clustered into 4 groups with very different shapes of trajectory. Like the first group, it kind of have a valley during this A/A age range, and it was enriched of like peptide chain elongation pathway and some other. And the second group is kind of monotonely increased with age. Third group is monotonely decreasing with age. And the third group kind of has a mountain shape with the highest abundances during this A/A age span. So one curious question is, okay, we see those trajectory shapes in the tumor. They look similar to those trajectory in the normal brain tissue, or actually this is something very unique to the tumor tissue. So to answer that question, we leveraged 2 data sets of normal brain tissue. One is an RNA data set from the brain span study, and the other is a proteomics data set from actually a study from Sinai team. And based on this 2 reference data set of normal brain tissue, we will be able to estimate the developmental trajectory in the normal tissue and compare that with the developmental trajectory in the tumor tissue and trying to see, okay, whether there is a significant distance. Okay, so this figure is a summary about contrast to the tumor developmental trajectory, tumor DTT versus what we estimated based on the normal tissue. And the left side is the result for male patient, for male, and the right side is for female. And we definitely see, even by eyeball, that yes, the trajectory in the tumor and normal are different for subsets of proteins. And especially as I highlighted here, this black box, we can see that most of the protein in this region has a monotone increased trend in the tumor tissue, but in the normal tissue, it actually has a increase and then decreased shape. And if we do pathway analysis, enrichment, actually, those proteins are enriched in, for example, the oxidative phosphorylation and a few other pathways. So actually, by using this strategy, we identified a collection of pathways showing significant tumor normal differences in terms of their developmental trajectory. So that really gives us a lot of insight. And then I want also quickly touch base on other orthogonal part, which is the residual part. So again, that is the information in the molecular data after we remove the developmental impact. So if we take that RDT matrix, which is basically this heat map, each column is a patient, each row is a protein. And we can clearly cluster into four groups of samples, 4 groups of tumors, as well as 4 groups of kind of proteins. And so here we are actually working on a harmonized data set of both HOPE and GBM, adult GBM data. And okay, I want just to say one more sentence. I know the time is up. Is that amazingly that we found actually those subtypes showed a very consistent concordance with the molecular subtype that has been found in the GBM domain. That's basically the proneural, mesenchymal, and the classical subtypes. So this finding actually is very controversial, and it's kind of surprising to us as well. We are still looking into more about this, but the excitement is that seems that if we are able to really dissect the developmental aspect in those molecular profiles, we start to see more consistency in the tumor across different age span from pediatrics, FA, and adult. And if that is real, that can be validated, then probably this can open some other new thoughts and thinking about how we can borrow information from different age group to better treat those patients. And we still, actually, this is a big project. We still have a lot of data that we want to look into and integrate. And in the end, again, this is a great teamwork, and really thank the leadership, and also want to thank the patients and the patient family who has contributed tissues to make this project feasible. Okay, I will stop here.